Okay, so we're going to continue with chapter 15. This is we're going to start on the section on protists. So we talked last Thursday about bacteria and about prokaryotes. So that was the yellow section here mostly. We kind of are ignoring archaea. I know archaea are covered a little bit in your text, but they're prokaryotes. They're I don't know, it gets confusing if we try to cover all prokaryotes, so I'm just going to focus on the bacteria side of things, and we'll leave it at that for that. This chapter's already broad enough. So we won't focus on archaea, but what we're going to be looking at today are organisms that are eukaryotes, and we're going to start talking about eukaryotes. Eventually, we're going to end up talking about all of these. Next chapter is going to be about land plants and fungi, which are two of the red lines up there. And then after the next test, we'll have an entire chapter on animals. So today, we're going to talk about all the gray lines that are in that blue circle, basically. So still an incredible amount of diversity. And this chapter covers an incredible amount of diversity. It covers the yellow and the peach circles, as well as all the gray lines in the blue circle. So it's an incredibly diverse chapter. I'm going to boil it down to just a few key aspects of it. But it's kind of important to note before we get into any details or any kind of concepts of the eukaryotes and of the eukaryotes that are not fungi, animals, or land plants, is to talk a little bit about how eukaryotes are different than prokaryotes. And the major thing that makes them different is the presence of membrane on the inside of the cell. And your book goes into a little bit of detail of how we think this evolved, but the main idea is that ancestrally everything was a prokaryote. So prokaryotes were the first organisms that existed on Earth. At some point, a prokaryote had its plasma membrane, the membrane surrounding the cell, fold in on itself. So there was a prokaryotic cell that at some point, part of the plasma membrane folded in and eventually pinched off. And this membrane would pinch off and form internal cell membrane, and the DNA somehow got put into this, so you ended up with DNA inside of this. And that created the first nucleus. And the same thing would have happened for the endoplasmic reticulum, the Golgi apparatus. Every other membranous body would have originated by pinching off of the cell membrane. So something pinching off and then making one of those internal cell structures. So that's the thought of how the endomembrane system, so the nucleus, the ER, the Golgi, lysosomes, things like that, the vacuole in a plant cell might have come to be in the first case. So through what is called membrane infolding. But really what I want to spend more time looking at, and we're going to look at this again later on in this chapter, is where mitochondria and chloroplasts came from. Because those have a different origin. They're not just bits of membrane folding in and the cell now acquiring membrane on the inside of it. They're actually entirely separate organisms being eaten by an early eukaryote. So you have an early eukaryote that has some membrane inside of it, but doesn't have any mitochondria, doesn't have any chloroplast. It now needs to make ATP, needs to make energy of some sort. And it's still doing it the way bacteria did, which we haven't talked about. But what they used to do, or what bacteria do, is they actually use their outer membrane like a mitochondria. They pump hydrogen out into the environment, and then as hydrogen diffuses back in, it makes ATP through ATP synthase. So similar to what we've talked about in the past. This was not all that efficient, and especially as eukaryotic cells got bigger and bigger, and had more membrane on the inside of them, and they had maybe more energy demands, this was not very efficient. So at some point, a eukaryote got energy by eating a bacteria and not digesting it. So instead of this being its membrane that's coming in, perhaps it folds in a membrane, but inside of that is a bacterial cell. And so now it's got the bacterial cell membrane, and it's got its external cell membrane, and as it pinches off, it now has something that's starting to look like a mitochondria. It has those two cell membranes. And so now when the bacteria pumps hydrogen out, it's now pumping it into that gap between the old bacteria membrane and the eukaryote's cell membrane. It's making that bacteria much more efficient. So it's actually beneficial for that bacteria. And so the bacteria is able to make so much ATP that maybe it releases some and the eukaryote can use some of it. So it starts out as what we call a symbiotic relationship. The bacteria that was eaten by that eukaryote is providing ATP. So ATP comes out of that bacteria, and the eukaryote is providing this new habitat for the bacteria that makes it really efficient. And so many, many years passed, and soon, eventually, this eukaryote no longer has to eat a bacteria to get this started. It figures out how to, when it's dividing, when it's going through mitosis, to also have the bacteria divide. And so then that bacteria stays inside of it. And that's the first mitochondria. So mitochondria arose from a process called endosymbiosis. So they're having a symbiotic relationship with a bacteria that they've basically eaten. So they've taken in a bacteria and have a symbiotic relationship with it. Does that make sense, how that might form a bacteria? You don't have to know any of the details of how it actually functions and how it went from eating a bacteria to reproducing with the bacteria. That would be something that you would learn if you started taking an evolution course or something like that. But that's the idea of endosymbiosis. And there's evidence of it still around today. The fact that mitochondria have two cell membranes is evidence of that. We think that chloroplasts did the same thing. So you start out with a photosynthetic bacteria, and the photosynthetic bacteria is eaten by a eukaryote, and it keeps that photosynthetic bacteria inside of it to produce sugars. And again, it provides a house for the bacteria, and the bacteria provides sugar to the eukaryote. So it starts out as this symbiotic relationship, and then ultimately they're able to divide together, and those are your early photosynthetic eukaryotes. So we'll talk about this a little bit later on, but that's the idea of endosymbiosis. And right here, where this arrow points to, is where we think the mitochondrial endosymbiotic event occurred, because mitochondria look pretty similar in all eukaryotes. So it must have been a common ancestor to all modern eukaryotes that engulfed that bacteria and kept it inside of it to make it into the first mitochondria. So mitochondria arose here. We'll talk about where chloroplasts show up on this tree once we get to photosynthetic organisms. We start talking about algae as we get along. Okay, so endosymbiosis is the mechanism by which mitochondria and chloroplasts would first evolve in eukaryotes, early eukaryotes. Okay, so all eukaryotes have membrane, all eukaryotes have a mitochondria. What the heck is a protist? Because that's what this chapter is titled. It's titled Bacteria and Protists, basically, is what I've called it. Protist is not a evolutionary, not a taxonomic, systematic, really, classification. It's a catch-all category, and it's defined more so by what's not in it than by anything else. It includes eukaryotes that are not plants, not animals, and not fungi. So plants, animals, and fungi, they all have their own kingdom. Protists are everything else that is a eukaryote. So we used to call it the kingdom Protista, but we've since learned that there's a lot more to it than that. It's not really a kingdom, and I'm going to show you this in here, but you can kind of see it along here. Here are all different kind of kingdoms, if you really or they'd be kind of super kingdoms, I guess we'd call them. Excavata, Chromoalveolata, Rhizaria, Archaeoplastida, and Uniconta. And you can see all the things that are yellow are protists. And you'll see that land plants, which is in the Archaeoplastida, and fungi and animals, which are in Uniconta, are on there as well. So they don't really form their own thing. So you'll notice that these yellow things are not a single clade. It's many different clades. And if you remember what a clade is organisms that include their most recent common ancestor and all of its descendants. So eukaryotes is a clade, but protists is not, because land plants, fungi, and animals would all be in that same clade. You couldn't separate them out. So we're going to talk about them. Let's talk about some of their similarities. They're really a group based on the fact that they are eukaryotes that are pretty simple in structure. And that's why we used to think they were all in one kingdom, because just looking at them at the surface level, they have some similarities. So we will talk about them from that perspective. So protists, because they're from so many different kingdoms and because evolutionarily they have so much diversity, there's a lot of diversity within protists. There are protists that are autotrophs. So there are a lot of algae, and we'll go into some algae later on and what that actually means. That's also a catch-all term that has things from different kingdoms. So here's Caulerpa. It's an autotroph, which means it photosynthesizes, makes its own fuel. You have heterotrophs. You have parasites. So this is a trypanosome, so it's a parasitic protist. And then you have ones that are mixotrophs. So mixotroph is something that photosynthesizes, but it also will eat things occasionally. And Euglena is a mixotroph. It can photosynthesize like autotroph, like Caulerpa here, and it can feed on other organisms like heterotroph, like trypanosome as well. So because there's so many different types of protists, I'm going to boil it down to just 3 categories, and then we're going to talk about those 3 categories just to really simplify things down. And your text has done a pretty good job of simplifying this, considering you could write many books on just this group of protists. So we're going to group them by lifestyle. So the first thing we'll talk about are protozoans. Protozoans are protists that get their energy by ingesting food, so they are heterotrophs. Then we're going to talk about slime molds. Slime molds, they resemble fungi, which we'll get to in the next chapter. But this is all due to convergent evolution. Convergent evolution are 2 things that look similar. We talked about this in the last chapter. They're 2 things that look similar, that have similar traits, but not due to shared evolutionary past. It's due to similar pressures, similar evolutionary pressures. So having the same kind of selective pressures can lead to 2 things having similar traits. So this would be like wings in bats versus wings in birds. They're both wings, but they have different evolutionary pasts. You have birds, or so like a seagull, which is a bird, and then you have a bat, which is a mammal. They have different pasts, but they both have wings. So slime molds resemble fungi, and they also have similar kind of roles as fungi. They are decomposers, and we'll talk about what that is. And then the last group we'll look at are algae, and algae are all the photosynthetic protists. So they are unicellular, they're colonial, they're multicellular, like our seaweeds, which is what I study. So there are many different types of organisms, but algae is kind of a catch-all term for all protists that happen to be photosynthetic. So we'll spend some time on that because that's what I know the most about. So we'll do that. Okay, so that's really the outline of what we're looking at today. I'm going to go through each of these groups, tell you a few features of each of these 3, and that'll be kind of our introduction to protists. Okay. Okay, so the first one, protozoans, protists that get their energy by ingesting food. So protozoans, again, is a functional group. It's a group that is based on similar kind of traits and similar feeding styles. It's not at all an evolutionary group. So protozoans are all the boxed lineage here. So we have 2 protozoans in the Excavata kingdom. We have 2 in the Chromoalveolata kingdom. We have 2 different groups of protozoans in the Rhizaria kingdom. And we actually have 4 in Uniconta, which is the group that humans are in. So we're in the animals and fungi are in Uniconta as well. So there's a lot of genetic diversity, a lot of evolutionary diversity within protozoans, but they all have similar characteristics based on their feeding mode and their mode of life. So we're going to talk about their similarities more than their differences. So protozoans are also called animal-like protists because they are heterotrophic, because they don't manufacture their own food. They have to go out and eat things. They can be unicellular, or they are unicellular, though. So unlike humans, they are all single-celled organisms. So they're unicellular and heterotrophic. And hopefully you all know what heterotrophic means by now. It means that they eat things, whereas autotrophic would mean they create their own food. Some protozoans live as parasites in animals as well. So here's some examples of protozoans. Giardia, which is up on the left there. Giardia is found in drinking water and drinking water that's been contaminated with feces, and it causes severe diarrhea. So this is often what gets people when they go to countries that don't purify their water very well, or if you drink unpurified water. So if you go out into the woods and you just drink from any stream, it might have Giardia in it because animals poop everywhere, and they could poop in the stream, right? So if you are out in the woods, you tend to look for headwaters, you look for springs, you look for early sources of water that haven't been contaminated by feces, that don't have Giardia, or you bring something like iodine with you so that you can kill off those things. But that's what would make you sick if you drank contaminated water, is Giardia. The middle one, Trichomonas. This is a STD as well. It's a sexually transmitted parasite, and it will give people urinary tract infections as well. So it primarily invades that. So it's another flagellate. It has a flagella in it. Amoebas, which are on the right. Amoebas are flexible. They move by changing their body shape. They cause something called amoebic dysentery, which causes about 100,000 deaths per year around the world. So that, again, you can get from contaminated drinking water, and the amoebas are going to cause pretty much everything that contaminated water causes, which is vomiting, diarrhea, dehydration, and that can lead to death, things like that. So very pleasant, a lot of these things. Forams, which are on the left here, that's a foram. Forams are very similar to amoebas, but they have a really hard outer shell to them, so they're really pretty looking. They've got these kind of spiky outer shells to them. Otherwise, they're pretty similar in structure to an amoeba. Apicomplexans here. Apicomplexans, that name means complex apex or complex top to it. And it's got this pretty elaborate top to the cell, which, and it's kind of shown over here, this is actually apicomplexans infecting a red blood cell. The complexity to that top of the cell allows it to attach to and kind of burrow into or penetrate its host. This is a species that causes malaria. So can be carried by mosquitoes in certain parts of the world. So South America, parts of Africa have this as well. And if you get this, it will infect your red blood cells. It causes a lot of the same symptoms as the flu, but so malaria is not a virus like Ebola, like that. It is actually a protist. So it's actually a protist that's infecting you. And the last one here is a ciliate. Ciliate have cilia, and we talked about that a long time ago, the little kind of hair-like things all over it, and that's how they move around. So they move with cilia. These are parameciums. This is a cilia called a paramecium, and if you go out to one of the ponds on campus, you take a few drops of water and you put it under a microscope, you're going to see a lot of ciliates. They're very common in pond water. All right, so those are some just examples of protozoans. And really the take-home message for protozoans are animal-like protists, which means they are heterotrophic. They tend to be unicellular. They can act as parasites, and they can cause lots of diseases. But other than that, they are eukaryotes that are heterotrophic and unicellular. So that's what makes them different than animals. They are unicellular. Okay. That's the first one. So the second one is slime molds. And slime molds are super cool. And I've got a video that I'm going to show you on the next slide about slime molds. And hopefully the audio is what I think it is. So we'll see. Slime molds are decomposers. And decomposers will feed on dead tissue. So they will move around until they come across a dead organism, so plant or animal, and they will break down that dead material, and they'll get their food from that. The cool thing about slime molds is that they are unicellular, but they also kind of act like they're multicellular at certain times of their life. So they will be unicellular and solitary when they're feeding. So when there's plenty of food around, there's lots of dead organic matter around them, they're going to be a amoeboid and a unicellular organism. And that's what number one is, and that's under a light microscope. When food sources run low, they will colonize. So they will clump up, they'll cluster up into a big colony that starts to look like a slug, basically. So there's this big kind of solid structure, and that will allow them to move much faster. So as a single organism, it would take them forever to go from this side of the table to the other side of the table to search for new food. But as a colony, collectively, they can move a lot more rapidly. So they'll move into a colony, and that'll allow them to move around and search for more food. If they're still having trouble finding food, they might form a stalk for reproduction, and they'll actually work together to create this really tall stalk, and then the cells that are at the top of the stalk will reproduce and produce spores, which will then be kind of shot out from the stalk to allow them to disperse. So this way they'll be able to move to entirely new environments, or at least their offspring will be able to move to entirely new environments. So happy moving around feeding, single. Not so much food, and they want to move a good amount of distance. They form a colony. Starved of food, they will reproduce. They will form a stalk and reproduce. So very cool kind of structures that they do. So I'm going to show you a little video of this, and it's actually a video of them taking on these three behaviors with a narrator. So check this out.
These are tiny mixamoebae of the cellular slime mold Dictyostelium. When food is plentiful, the mixamoebae are completely independent as they feed and multiply. When their food supply dwindles, their behavior changes remarkably. When they become crowded, they start moving together from all directions, flowing into growing masses containing thousands of cells. Aggregation starts when one cell spontaneously secretes a pulse of a hormone called cyclic AMP. Nearby cells respond by moving towards the hormone and by emitting their own pulse. Soon this response becomes widespread and synchronized, creating waves of accelerated movement passing through the migrating cells. These mixamoebae are streaming steadily towards a distant aggregation center. As always, there are a few stragglers rushing to catch up. The aggregations of cells round up, and then each turns into a slug called a pseudoplasmodium. These slugs are active, searching the terrain for a suitable place to settle and leaving a trail of slime as they go. Remember that each slug consists of thousands of free-living amoebae which now have organized themselves into a cohesive unit and which synchronize their activity. During its travels, an individual slug may fuse with other slugs it happens to encounter. Meanwhile, a few individual mixamoebae are still visible, moving around on the surface. After this migratory activity, the slug rounds up again, but this time it flattens onto the surface and then grows a thin stalk with a fruiting body on the tip. Throughout these maneuvers, the mixamoebae remain separate individuals. During this final phase of reproductive activity, the behavior and fate of each cell continues to be subordinate to a self-generated pattern. The mixamoebae in different parts of the pseudoplasmodium differentiate into three main types of cells with different functions. Some cells form the base and others the stalk, while the rest migrate up the stalk and turn into spores in the fruiting body. The mature spores are later released.
All right, so pretty cool, I think. It's amazing when you think about what that is. So it's individual, single-celled organisms that, when food becomes scarce, they clump up and form a slug, basically, and then that slug moves around, and then they differentiate into some of those individuals playing a role as the base of the stalk, some being the stalk, and then only the ones that are at the very top that will actually form the spores and be dispersed. So basically the ones that form the base and the stalk are sacrificing themselves so that some of their relatives, which are in the tip of the stalk, can be dispersed and possibly survive. So it's kind of an odd thing when you think about it. They're individual organisms, but they work together so that some of them can survive those harsher environments where there's less food. That's what they do. So slime molds, decomposers, can function as a colony, can move as a collective. Okay. The third group, and the group that I'm going to spend a fair amount of time on because I find them the most interesting and they're what I know the most about, are the algae. So algae are photosynthetic protists. In Carolus Linnaeus' time, so you remember Linnaeus, he was the father of the kingdom, phylum, class, order, family, genus, species, so classification. He actually called all photosynthetic organisms vegetabula, and he had 25 classes of them, including cryptogamia for plants without seeds and flowers, and within that there were algae, fungi, muci, and fasciles. So algae were a member. So they already were talking about algae down in the 1700s. But it didn't take long after Linnaeus, who grouped all photosynthetic protists together into one group called algae, to find out that there's actually a lot of diversity in them, and these organisms fall into many different kingdoms as well. So what are algae? Algae are photosynthetic protists. So just like other protists, we often classify them by how they gain their food. So whereas protozoans are heterotrophic protists, algae are photosynthetic protists. So they reproduce partly or entirely by unprotected spores, but that's something, don't worry about that. They don't have seed. What that means is they don't have seeds, they don't have flowers, they don't have other complex organs like land plants do. And when we get to plants in the next chapter, we'll talk about the development of those things, the development of fruits, development of seeds. and all these protections for your embryos, for your offspring. Algae don't have those. They're very, very simple. They have a plastid, like a chloroplast, and they get that plastid from either primary or secondary endosymbiosis. So what we've talked about so far, when I talked about the mitochondria and the chloroplast being very similar to this is something called primary endosymbiosis. I'm going to tell you the story of what secondary endosymbiosis is as well, and why this actually makes things really hard for us, those of us that study seaweeds and study algae, because of the multiple different origins of chloroplasts. And algae can be single-celled, but there also are some algae that are multicellular. I'm going to talk about that as well. So there are some slightly more complex individuals. Not all protists are single cells. There are multicellular protists. They just happen to be aquatic plants, aquatic organisms. Okay. So algae is not a monophyletic group. And what I mean by that is not one single clade. So if you remember back to when we talked about clades, clades are groups of individuals that have one common ancestor, and it includes all the descendants. So just like protists are not monophyletic, algae themselves are not monophyletic. They're actually found throughout the eukaryotic phylogeny. So they're found in many different branches, and I'm going to show you a picture of where all the algae are found on that graphic of all eukaryotes. Many are closely related to some of those heterotrophic lineages. So a lot of those parameciums, those were actually very closely related to algae, and there's some heterotrophic lineages that are more closely related to algae than algae are to other algae. So algae is, again, a functional group. And this is the word functional group means a group that have a similar function, a similar body style, a similar way of gaining energy, but they're not based on shared evolutionary history. All right. So, before I start showing you some of the diversity of the algae, I do want to spend a little bit of time talking about what secondary endosymbiosis is, because it's a pretty crazy thing that's happened in these organisms. And endosymbiosis itself is a pretty crazy thing that's happened. So, okay. So first off, primary endosymbiosis occurred more than once. So we talked about mitochondria, so a heterotrophic, aerobic prokaryote, like an aerobic bacteria, was engulfed by a eukaryote, and then you have now an ancestor heterotrophic eukaryote, and that's probably what the ancestor to all eukaryotes looked like. It had a mitochondrion, and it had some internal cell membrane that it created through engulfing its own membrane. So then at some point, some organism took on a photosynthetic prokaryote. So mitochondria are the product of endosymbiosis with a heterotrophic prokaryote, so a eukaryote that eats sugars. And the plastid, the chloroplast, is a product of one of these heterotrophic eukaryotes taking on a photosynthetic prokaryote. So it brought in a bacteria, cyanobacteria perhaps, and kept it on as a chloroplast. So where we think that happened, if we look at the evolutionary tree, is we think it happened at the base of the plantae. So this group here, which I called Archaeoplastida on that picture, we call that by the kingdom name Plantae. And that includes land plants. It includes some algae. It includes red algae and green algae as well, and we'll talk about that kind of later on. So primary endosymbiosis of the mitochondria occurred at the base of all eukaryotes. Primary endosymbiosis of the chloroplast occurred at the base of the plant kingdom. So plants, red algae, and green algae are all got their plastids by what's called primary endosymbiosis of a photosynthetic bacteria, some sort of photosynthetic prokaryote. So, some point after this formation of the first chloroplast, red algae and green algae diverged. So green algae would ultimately be the precursors to land plants. So originally everything was aquatic, so we think, and green algae, in a process that we'll talk about in the next chapter, ultimately began colonizing land, and we'll talk about how mosses and ferns and trees have all evolved from green algae in the next chapter. And red algae have remained marine, and some green algae have remained marine or freshwater. So there was a split at some point. And both of them have plastids, both of them have chloroplasts that came from primary endosymbiosis. So if you look at the plastids of a red or a green algae, we call them primary plastids. And some of this information is just extra stuff that I'm throwing in here. I'm not going to test you on the details of a plastid. There's already enough details in here. But I think it's kind of interesting, especially when we start talking about secondary plastids. Primary plastids have 3 membranes. So they have an outer one that's from the host cell, and they have 2 inner ones from the bacteria that they engulfed. So as the membrane folded in, the bacteria that they engulfed actually had 2 membranes, and so they have 3 membranes. And this is what most land plants look like. This is what green algae and red algae chloroplasts look like. They have 3 membranes. There was a 4th membrane that was lost at some point during the evolution of these organisms, and it's the same in red and green algae. So what happened then is the really interesting thing, is that some organisms that had split off from that original eukaryote that were heterotrophic lineages actually had an endosymbiosis event where they took on a red algae or a green algae. So they engulfed a eukaryote. So they engulfed a red alga or a green alga. And this is called secondary endosymbiosis because it's engulfing an organism that engulfed an organism, basically. So it's taking on a eukaryote rather than taking on a prokaryote. So red algae chloroplasts, and actually red algae, are found inside of things like dinoflagellates, apicomplexans, and straminopiles. And green algae chloroplasts, green algae endosymbionts, are found inside of euglenids, so euglena, and chloracneophytes. So none of these do I expect you to remember these names, but we're going to talk about these groups later on and some of their characteristics. So what is really cool about this is if you look at these groups, some of them not so much anymore, but if you look at some of them, including the chloracneophytes, because the chloracneophytes seem to have retained the most of their green alga endosymbiont, you can actually see a nucleus. So they have these things called nucleomorphs, is what they call them. So if you look closely at the chloroplast of a chloracneophyte, it actually has an extra cell membrane, because it has the outside membrane of the green alga, as well as the plastid membranes. And between the plastid and that extra membrane, there's a nucleus in there, and they find nuclear DNA. They can actually find out which type of green algae was taken on through endosymbiosis. So they think there was actually 3 other secondary endosymbiosis events. One that led to dinoflagellates, apicomplexans, and straminopiles, one that led to euglenids, and a different green alga that was taken up by the chloracneophytes. And so by looking at the DNA of the chloroplast and that nucleomorph, if they can find it, they can researchers have actually been able to tell which green algae and which red algae was engulfed. And it's kind of neat that you can do that. You can actually see a nucleus, an extra nucleus inside of some of these organisms. So you won't find that in land plants, because again, land plants come straight off of green algae, and they had primary endosymbiosis, but some of these protists have secondary endosymbiosis, including the branch that I studied a lot, which was straminopiles there. So I did a lot of work there. So if we look at the tree, again, of all eukaryotes, the big green chloroplast shows primary endosymbiosis, and that was what created plastids in all the Plantae group, so which includes red algae, green algae, and land plants. And then there is a green algal endosymbionte for that chloracneophytes. A different green alga was taken up by the euglenoids over there, and red algae was taken up by dinoflagellates, apicomplexans, diatoms, golden algae, brown algae. Probably, if you're looking at this, it was probably two different events as well for the red algae, probably one for the straminopiles, which are the diatoms, golden algae, and brown algae, and maybe one for the dinoflagellates and apicomplexans. You may say, I thought you already said apicomplexans before. Apicomplexans is a heterocont. It's actually lost the photosynthetic ability of that plastid. It retains a plastid, but the plastid is no longer photosynthetic. So there's all kinds of crazy things, none of which I'm going to hold you to, but there's a lot to learn here. If you really wanted to go into protists, you could. I think I actually took 3 courses on protists when I was an undergrad. So that's just scratching the surface of these things. Okay, so all these groups that are not land plants, so all of the ones except for land plants that have a plastid either through primary endosymbiosis or through secondary endosymbiosis are called algae. So you can see why studying algae is actually really complex. I look at colleagues of mine that study one species of animal, and you can see where animals are on this graphic way over there. They're one teeny little branch, and they're actually looking at one teeny little branch off of that one teeny little branch. Whereas my degree, we studied everything that is circled. So a lot of diversity, which means you need to know a little bit about a broad range of things. And you can't actually study algae in a lot of depth. You have to study red algae or green algae or brown algae. So my group of work was in the brown algae, which you can see is in the Chromioalveolata. They're actually a complete separate kingdom from red algae or green algae, which are more closely related to land plants. So my group that I studied was actually as closely related to land plants as it was to animals or fungi. And actually fungi are much more closely related to animals than they are to plants or other algae. Okay, so I'm going to go through a few groups of algae and talk about them and some of the kind of the key things, the key characteristics of it. Pay attention to that outline that I posted on Canvas about this page for the aspects of this that I really want you to focus on when you're studying, so that you don't get bogged down in a lot of the details that I might not test you on. So I tried to make that sheet off of what I'm going to ultimately put on an exam, and I also gave you review questions of things that I'm going to put on the next exam, just to keep you from getting too broad in what you focus on. But I figure I can talk about a little bit more than I test you on here, because it's really interesting. Okay, so the first group that we're going to look at are some unicellular and colonial algae. And I've picked groups that either I really like, or they're groups that you might like or might already have heard something about. So we're going to talk about 2 groups of unicellular colonial algae, including dinoflagellates and diatoms. So dinoflagellates have these cellulose plates surrounding the cell. So just like plants have formed their cell wall with cellulose, dinoflagellates have this as well, and they have this kind of plate-like appearance. They're very well protected on them. They're a single cell, and they're surrounded by a cellulose plate. Diatoms also surround their cell. They're a single cell, and they also surround their cell with protection. But in diatoms, their protection is composed of silica, which is what glass is made out of. So they're basically a glass shell on the outside of the cell. So dinoflagellates have a cellulose shell on their outside. Diatoms have a glass or silica shell on their outside. Okay. So dinoflagellates, let's see where I am. Okay, good. We're doing well. Okay, so dinoflagellates, if you've ever heard of a harmful algal bloom or red tide, most red tides are a result of blooms or incredible increases in reproduction and biomass of dinoflagellates. So what happens is there will be a favorable set of conditions that causes a rapid increase in the dinoflagellate population. So here, it's usually runoff. So fertilizer, farms maybe laid down a whole bunch of fertilizer or manure or something like that that has washed into the Caloosahatchee, and then there was a lot of rain, or there was a release from Okeechobee, and it washes all that fertilizer out into the ocean, and it's now basically taken all these nutrients and flushed them into the Fort Myers beach area, and then all of a sudden, these dinoflagellates bloom like crazy. And a lot of dinoflagellates have reddish color to them, and that's where we get the name red tide, because you'll see this murky kind of reddish color. There are other things, like brown tides, things like that. They're all other species that have bloomed. These can be very harmful to humans. They can also be very harmful to marine life, because some species of dinoflagellate will produce toxins. So that's one thing. Also, just the fact that there are these huge biomass of the single-celled organisms, fish that are trying to get oxygen from the water, as they suck in the water, they're sucking in all these single-celled organisms that get stuck in their gills and can clog up the fish gills and deplete their amount of oxygen that way. Also, as algae bloom, they don't do photosynthesis as rapidly as they do cellular respiration, and so they start pulling oxygen out of the water. And so just the oxygen levels in the water even drop down, and you end up with a lot of fish kill. So fish will just float up to shore dead due to red tide. And if there are toxins in this, you can get toxins accumulating in shellfish, so clams or oysters that might be in that environment are sucking in a lot of these toxins and filtering them, and so it can be very bad for fisheries as well that are nearby. Even if it doesn't kill them, it can make the shellfish unsafe for human consumption. So dinoflagellates often cause harmful algal blooms. Here's one species of dinoflagellate that you may have heard of it. Carina brevis is a very common one that is related to these red tides. So there's certain species that tend to bloom like this more often than others, and especially they're well known if they produce toxins. Carina brevis produces a neurotoxin that can lead to massive fish death, fish kills. Closures of shellfish industries, again, through them filtering this toxin through them. And if you go swimming and you take in a lot of the water, you can get respiratory infections, respiratory irritation in humans. So here's an example of a fish kill. So you'll see all these dead fish floating and washing up ashore during a red tide event. It's probably because it's Carina brevis. And we'll get things like that down here. That's when they close the beaches, like that. Okay, I think this is actually a picture from Texas, which is close enough. It's pretty similar. They get similar species as we do. Okay. All right, dinoflagellates also have positive things that they do. They're not all evil. So reef-building corals, they have a symbiotic relationship with a dinoflagellate called zooxanthellae. So they actually have dinoflagellates living inside of the coral, and the coral provides protection for the dinoflagellate, keeps things from eating it, and the dinoflagellate provides food sources for the coral. So if you've heard of coral reef bleaching before, anyone heard of that? So it's where the coral reefs just all start turning white. The reason why they're so colorful is because of the algae living inside of them. When coral reefs bleach, what's happening is they're losing that endosymbiont. They're losing the dinoflagellate. Often it's from heat stress, but it could also be from other things as well. Sorry about that. Could be from other factors as well, but it's often from heat stress, either due to climate change or just due to there being freak kind of high heat environments in the coral reefs. And so the zooxanthellae leave, the coral turn white, and often the coral will die because they need those nutrients coming from the dinoflagellates. All right, so positive thing as well for dinoflagellates. Okay, the other type of single-celled algae that I'm highlighting is a diatom. And diatoms, if you remember I said, have silica shells to them, so glass shells to them. You may have heard of diatomaceous earth. Diatomaceous earth. That is diatoms, basically, that have been fossilized and then crushed up, and is a very, very nutrient-rich soil that we often will use for planting things in. But there are things like fossilized diatom remains that form these, the White Cliffs of Dover are all diatoms, if you've heard of that before. We also use it for many useful human things: insecticides, filter material, kitty litter, even dynamite has diatoms in it as well. So it's really, so filter material, it's very fine grain, it's like sand, but it's silica rather than sand, and it can filter. You can actually have diatomaceous filters for pools that exist. And then kitty litter will have diatoms in it. That silica sand material is actually really absorbent as well, so it will absorb waste, things like that. Okay, so diatoms. Diatoms are also, one of their biggest importance is their ability to fix carbon. So diatoms are massively abundant. They're teeny, you don't see them, but they're basically in any aquatic environment you'll find diatoms. So they have huge ecological importance. They account for about 20% of global photosynthetic fixation of carbon. So they pull out 20% of the carbon dioxide that's taken on by any photosynthetic organism. And that number should be impressive. It's more than all of the world's tropical rainforests combined. So if you add up all the diatoms on Earth, they have a much bigger impact than all of the world's tropical rainforests. So a lot of carbon is pulled up by them. They're one of our biggest kind of remediation organisms against excessive carbon that we are producing through burning fossil fuels, through a lot of our day-to-day activities that produce a lot of carbon dioxide byproducts. So they'll fix carbon. Actually, in the ocean, they fix carbon and then they sink, so they actually will pull it out pretty much permanently from the Earth as they die. So, all right. So I guess one way to think about this is that every fifth breath that you take is thanks to a diatom. So they provide, because they fix 20% of the world's carbon, they also provide 20% of the world's oxygen. So every 5 breaths you take, you can say, that was a diatom that gave me that breath. All right. Okay. Okay, so those were the common single-celled algae. Brown algae are an example of a multicellular protist. So brown algae are multicellular marine photosynthesizers. They are the largest as well as the most complex of the algae. They're much more common on temperate coasts than they are in the tropics, so we don't get nearly as many species of brown algae down here, and we get more than you would get closer to the equator in Florida. But you get way more up in the north. So where I was living in Canada, there was much more diversity of brown algae up there. They like colder waters. So down here, they'll be found pretty deep in the water, whereas up north, they're found more shallow. So this is a species called Palmaria palmiformis. So Palmaria palmiformis. Any idea why it was named that? Yeah, it looks like a palm tree, right? So it looks like a little palm tree. And the neat thing about it is that it does have some differentiation, just like a palm tree does here. It has what's called a holdfast, which, like roots, holds it to the rock surface. They don't have the necessity of getting water through their holdfast because they're actually submerged in water during part of their life. So this is actually an intertidal organism where the waves will kind of splash onto it, and that's where it gets its water from. So it has a holdfast. It has a stipe, which is a lot like a stem of a land plant, and it has blades on the top. the really cool thing that this species does, Palmaria palmiformis, is the way it reproduces. As it lives kind of right near the top, so if this is the rocky intertidal, there is kind of water here, and there'll be waves that come up. The Palmaria will be growing right here, right in what's called the splash zone. So they're not going to be completely submerged in water any time of day, but the waves will splash onto them. And when they reproduce, they actually release spores from the blades, and their blades are designed so that as water splashes them, it will wash the spores down, and they'll drip off the tips, and it'll allow them to kind of drop spores around their feet. And so they'll gradually expand out, and so they'll kind of take over this environment by using water to disperse them really short distances, because they want to stay in that splash zone. As they found, probably if they're too low in the water, they'll get eaten by something, and if they're too high, they'll dry out. So they want to stay right in that splash zone. So they have a neat way of adapting to be able to drop them all around them. All right. So some brown algae, so these guys don't do it, but there are giant brown algae. So these are things called macrocystis. They can grow up to 180 feet tall. So they'll be all underwater. So they'll make these underwater kind of forests, and they'll be called kelp forests. These guys actually are so big that they've even come up with a way to move nutrients around. So considering other algae are single cells, until we get to the red and the greens, they don't have the needs to move nutrients from cell to cell. Here, these things can be so big that the upper bits of them are really well exposed to light, but their holdfast way down at the ocean floor that might be 180 feet deeper than their leaves, might be pretty much in the dark. So they need a way to get nutrients, get sugars down to those cells that can't photosynthesize very well. So they actually, inside of them, have a series of tubes that they will push their nutrients down to the lower cells with. So kind of cool. They act a lot like land plants, and we'll talk more about how land plants deal with being on the land. These guys need to deal with maybe not being completely exposed to light all the time. They are really, really important habitats for many, many animals and other organisms that live in this environment. So they provide habitat. If you look near the base of these kelp forests, there'll be lots of crabs, lots of fish, lots of other things that live on them. There's things that feed on them. There are these really rich environments. And when we get to the ecology section, we might talk a little bit about the relationship between kelp and sea otters and actually sea urchins. So there are sea urchins that eat kelp, and there are sea otters that eat the urchins, and they actually all work together to create this nice balance. And we'll talk about what happened when otters were hunted for their furs, what happened to the rest of the environment, and the diversity of organisms found there. They also will dampen the impact of waves and reduce shore erosion. So these waves that are coming in offshore, down here, there might be these giant kelps, and because of them, the surge of water coming this way is dampened down, and there's not as much wave action on the shore, so the shores don't get eroded as much. So they actually help prevent that erosion a fair amount. Really cool thing about brown algae and these kelps is something called alternation of generations, and we'll talk more about this again in the next chapter when we look at this in plants, but I want to introduce it here. Alternation of generations is a concept that organisms can have more than one life stage. And in the case of brown algae, they have two multicellular stages. So we've talked about this briefly in terms of reproduction of humans. When humans go through meiosis, what cells are they producing? So you've got a human, and you've got a human that's female, all right? What cells are they producing through meiosis? Yeah, so sperm cells and egg cells, right? So these are what are called N, or haploid. And then they come together and they produce a zygote that is diploid, or 2N, right? And then the zygote will grow into one of these organisms. It's also diploid. So humans have only one multicellular stage. They are diploid. So this is a diploid and multicellular, whereas here it's single cell and haploid, right? So seaweeds aren't like this. They actually can be multicellular in both steps. So this large organ, and I'm going to show you this actually in the next slide so I don't have to draw it. You don't have to look at my drawings. So the large bladed kelp, which is diploid, will form spores. So it'll undergo meiosis, and it will make these haploid spores, single cells that are haploid. It will then release the spores, which are called zoospores. Those spores will land somewhere, and they will start to divide through mitosis. And they'll create something called a gametophyte. And a gametophyte is a haploid multicellular organism. So it's haploid, just like, it would be like if males release sperm, and then that sperm started growing into a multicellular organism somewhere. Really kind of gross, right? But that's the same idea, right? So it's as if sperm didn't first look for an egg. Sperm actually grew into a multicellular organism. And that's called the gametophyte. The gametophyte, there'll be a male and a female version of the gametophyte, so it'd be like the egg would do that as well. These would grow into multicellular organisms. And then at some point, they would release spores that they produced just through mitosis. They don't need to have the number of chromosomes again. They come together and they form a zygote. There you go. And the zygote then develops into the diploid sporophyte again. So in this case, for a lot of kelp, they'll actually have a large structure, which is the sporophyte, and they'll also have a small gametophyte. But it's not always like that. There's a type of brown algae called a Ectocarpus. So that's the genus name. And Ectocarpus is a filamentous brown algae. And it actually looks kind of, it's not a very good marker, but it's just this branched species like this, and it'll be 2N. This will be your sporophyte. And then the gametophyte looks exactly the same. So they have alternation of, they call them isomorphic generations. So the gametophyte and the sporophyte actually look exactly the same. So this would be like a male releases sperm and it grows into another whole human, male, that happens to only have one copy of each chromosome. So instead of 46 chromosomes, it would be a human with only 23 chromosomes. And then at some point, they actually do the sexual reproduction part of it. They actually release sperm that then finds an egg, which came from a haploid female, and they come together. So really hard to imagine it in terms of humans, because we don't do it this way. We do what's called a diplontic life cycle, where we're only multicellular as a diploid. And there are some algae that do that, but most algae have a multicellular diploid and a multicellular haploid stage. And we're going to talk about this in plants as well, because plants do things along the same lines, especially with things like ferns and mosses. Trees are a little bit more like us, but ferns and mosses are a little bit more like the algae. All right. Okay, so this is called alternation of generations, and it's alternation between a haploid gametophyte and a diploid sporophyte. So you should recognize those words at least. So we have sporophyte, which is diploid, and gametophyte, which is haploid. And just to double check, you guys know what I mean when I say haploid and diploid? No? So haploid versus diploid, what does it sound like? The word hap and the word dip. All right. Sorry, so it sounds like single versus more double, right? And it's not cells, but it's chromosomes. So haploid is one copy of each chromosome, whereas diploid is two copies of each chromosome. So we are diploid. We have 23 pairs of chromosomes. We become haploid for sperm and egg cells. They have 23 chromosomes, 23 in sperm, 23 in eggs. When they come together, they form a zygote, which is diploid. It has 46 again, 23 pairs. So in this case, a sporophyte has pairs of chromosomes, whereas a gametophyte has single chromosomes. So it is half the number. So if the kelp as a blade had 40 chromosomes, the gametophyte would only have 20. It would have half the number. And then, so there'd be a male gametophyte with 20 and a female gametophyte with 20. They would then both release cells, which would come together and form a 40-cell sporophyte again. So the only difference between the human life cycle and the algae life cycle is that they can be multicellular when they are haploid, whereas we only exist as haploid as a single cell, as a sperm or an egg, and it's very brief, right? Only lasts for, sperm lasts for maybe 3 days, so you've never been haploid for very long as a human. But if this cell would divide, it would become a multicellular haploid individual, which would be called a gametophyte. And a multicellular diploid is a sporophyte. So you're going to hear these terms again in the next chapter, and that's why I'm introducing them now, is that I'll use them again. And we'll talk a little bit about different life cycles in different organisms besides algae. Okay? All right? All right, I'm trying not to make this too complex, but I get a little excited with the seaweeds, so I could talk about them a lot more, and I'm trying to just give you digestible amount of information. So please stop me if it gets too overdone. This is the last slide, though. This is the last I'll do. So we'll talk more about green algae next class when we start looking at the origin of land plants. Green algae and red algae are both members of the kingdom Plantae, so they do share a lot more similar characteristics to land plants than the brown algae or other protists that we've talked about. They have, remember, they're placid from primary endosymbiosis. We're not going to really talk about them in more detail. Again, that would be an entire course to go into details of some of the crazy things that red algae do. Green algae are land plants, basically, that are not on land. So land plants are most closely related to the green algae, and in fact, land plants are a branch off of the green algal tree. So if you look at everything that's called green algae, in order for that to be a single clade, you would have to actually include land plants in it, because there's so much diversity in green algae. So we categorize land plants as their own thing because they're on land and not in water, but in fact they do share a lot of similarities with green algae, at least at their origins. So we'll start on Thursday talking about land plants that are not drastically different than green algae, that are just barely on land. And then we'll work our way up to land plants like flowering plants that we see all around us that are very well adapted to land. So we're going to talk about how land plants have colonized the land over time. Okay, so hopefully that was a good introduction to protists. Protist, remember, is just a functional term, and there's a lot of diversity in it. Okay, that's it for today.